10 minutes. So I actually have 0 announcements today at all, except that the sun is out, and that makes me feel considerably happier than I have been for the last couple of weeks. We have a guest speaker today from Ann Arbor, Michigan, and she's been invited by the Division of Pulmonary Critical Care and Sleep Medicine. And so to give the formal introduction, I'm going to ask Dr. Saad, who is the division chief, to please come to the podium.
Good morning, everyone. It is actually our last pulmonary grand round, and we are very fortunate to have our guest speaker today. I'm pleased to introduce Dr. Yvonne Huang from Ann Arbor, Michigan, rising star in the field of academia if you look at her resume. I'll give you briefly a little bit about her. She went to college at Stanford and medical school at University of Alabama, and then she went to medicine residency at Yale and did her fellowship at University of California, San Francisco, and this is where she got interested in her area of career and microbiome. And so she stayed for 3 years and for fellowship, 2 extra years for research fellow, and then spent 4 years there working on starting her career in this area. And then she moved to Michigan in Ann Arbor. She's assistant professor there and continued her work in the area of microbiome, which is a hot area. And we know, of course, we have this in GI. She is well-funded, NIH funded, and regular invited speaker to CHEST and ATS. And in a very short time, he has numerous publications and awards. As I said, clearly a rising star in the field of academia and pulmonary. I'm very happy to have her here. And we're going to learn more about the relationship with the microbiome with the asthma. So thank you for coming and welcome.
Can you hear me? I don't think the microphone I'm wearing is working, but is it? Okay, I'll just use this one standing here. Thank you so much for the very kind invitation to come down to Louisville. I've actually, as you've heard, lived in many places, and I've driven through Louisville on I-65 and never actually hopped off to be downtown, so it's a pleasure to be here. And I'm very excited to share with you some information and data from the culmination of some of the work I've been working on for the last 10 years or so in the space of airway microbiome and asthma. And we're starting to do some work in COPD as well, which I obviously won't talk about today. But my goal is to give you a flavor of where the field of microbiome research and lung disease with a focus on asthma has come over the last several years. In the context of asthma, here are the objectives. I do want to spend a little bit of time reviewing evidence linking the microbiome in early life to childhood asthma. I realize we're all adult physicians here, but in the context of allergic airway diseases, some very brief overview I think is good context to have because we draw a lot of inference from the wealth of literature and the studies that have been done over the last 2 decades in the childhood pediatric asthma realm. And then I'll focus the middle of my talk about just telling you a little bit about recent evidence linking the airway microbiome to asthma phenotype in adults, and that's work that's I've been more directly involved in the last couple of years. And then finally, I would like to offer some discussion about challenges and new directions to understand how microbiota influence lung disease phenotype and some of the considerable challenges that still remain in doing this in the clinical context. So as many of you are familiar with, asthma is a very common, highly prevalent disease. It affects about 300 million people worldwide. And this is now an old slide drawing from data that the WHO published back in 2004, and that was recapitulated in a review article by Science in 2011. And I think the flavor I want to impart to you is just the idea that asthma is really much more of a first world disease. It's much more prevalent in westernized countries, industrialized nations, and even in large-scale epidemiologic studies over the years that have looked at populations where the demographics are changing quite a bit, such as parts of China, Africa, which isn't really captured here in the map. But as you have increasing levels of urbanization and people migrating from more rural communities into the urban environment, those growing urban centers see an increased rise in allergic as well as autoimmune diseases such as asthma. Now, asthma is not all the same, and I think that's the flavor of the phenotype definition that has emerged over the last 20 years or so, and become more and more refined with many large network studies funded by the NIH trying to dissect, at least doing some sort of clustering type analyses to find different asthma phenotypes. And that's what's captured up here in this top tier of this review article I did with my mentor a couple of years ago when we were at UCSF. And just to summarize, what we sort of view microbiota and the science of microbiome fits into studying lung disease. At least in the context of asthma, we know a lot about genomics of asthma, susceptibility genes. There's been intense interest in that, but really the function of a lot of those genes and how they impact disease phenotype remain a little elusive. We certainly know a lot about the immune system, the adaptive immune components that play a role in shaping asthma, and I'll discuss that a little bit more. A lot of data on epithelial and mucous cell biology. And in between, we feel like that microbiota, at least of the mucosal lining surfaces, such as the respiratory tract, have really been overlooked in a clinical context, and that's where we've focused our energy over the last couple of years. I think in the clinical context, certainly in the pediatric literature, this is well established, these environmental exposures in the very early life window that impact gut immune development through the microbiome can have a profound impact in susceptibility to allergic diseases later in life. But in adults, we recognize that these comorbidities listed here, and they're just one of several, also are important comorbidities in asthma. We know that significant sinus disease in the nasal cavity certainly can increase morbidity and symptom burden with asthma. Obese-related asthma is also an entity that's not well understood. We think of obesity as much of a systemic inflammatory disease, and we think that has a role in lung disease, but certainly the mechanisms are not really well understood just yet. And a gastroesophageal reflux disease has also been associated with increased asthma symptoms, although large trials looking at the role of PPI interventions and asthma control really haven't been negative, but clinically we recognize it as contributing to cough and symptoms of shortness of breath. So I think the latest definition from the Global Initiative for Asthma in 2014 really tried to highlight this variability in disease phenotype and heterogeneity there, and I won't read it, but that's highlighted in these yellow terms that were included in the definition of asthma that came out in 2014. So I wanted to spend just a minute or two just setting definitions straight, because I think the microbiome term has been so popular over the last 10 years, and in literature, when we review papers, I often see microbiome being referred to, and yet we're really talking about microbiota. And so what's the difference? This definition is borrowed from a lovely sort of perspective piece that was published by Marchesi and Ravel. Jacques Ravel is a microbiome scientist at the University of Maryland. I think he's actually editor-in-chief of this new journal that's really rapidly risen in impact factor. But he and his co-author here proposed a definition that microbiome really is not just about the organisms, and that's what the definition has been used loosely in the literature, and those of us who work in the space are trying to be more attentive to terminology. But it really encompasses the biome, the entire habitat, the ecosystem that includes the microorganisms, what their genes potentially can do and do, and the surrounding environmental condition or microenvironmental conditions. That's really the microbiome. And in the context of discussing the human microbiome, as a part of this National Academy Committee that was part of the last year and a half, we decided to make it clear that microbiome could be used in conjunction with specific sites. When we talk about the gut microbiome, the respiratory microbiome, the skin microbiome, or as an all-encompassing term to refer to all microbiomes on or within humans. So microbiota really is the more proper term when we talk about the actual organisms, and that really has been the focus of most studies to date. Surveying, cataloging, understanding what's different in a particular site or niche compared to a different patient group or a different site, and that's sort of where the technology and the wealth of evidence has been today, is focusing on the description and understanding of what types of microbes differ. And so these microbes largely have focused on bacteria to date, but obviously encompass fungi, viruses, helminths, and another tree of life out branch called archaea, which are more prominent in the gut, we think, than other body niches. But really, microbiota is a part of the definition of microbiome, and so we try to become more attentive to that. Now I'm going to move briefly into reviewing some of the evidence linking the microbiome in early life to childhood asthma as a setup to understanding what has informed our thinking about studying this in the context of adult lung disease. And this is an immense field, which would be a talk or class in and of itself, and so I certainly don't have time to go through all of it, but I wanted to show you highlights from studies over the last 10, 15 years in different spaces. But first of all, I wanted to highlight the idea that inappropriate responses to microbes, or how we handle responses when the body is exposed to microbes, is sort of embedded in some of what we do know about asthma susceptibility genes. And so this is a summary review that was published by Donato Vercelli, who's a geneticist out at Tucson a couple years ago now, and really summarizing all these genes that have been associated in GWAS studies or any sort of studies over the last 20 years being associated with increased risk for asthma. And they can be grouped broadly into 3 groups. I'll start down here with type 2 Th2 effector functions, which we think of most of all in the context of asthma as an allergic disease, but certainly not all allergic mechanisms. But there's certainly genes involved with type 2 immune functions that have been involved with that. And you see IL-4, IL-13, IL-5 showing up here. There's also polymorphisms in genes we thought to be involved with innate immune responses, microbial recognition, immunoregulation at the mucosal barrier. And that includes toll-like receptors, nod-like receptors, and other mucosal surface genes that have been associated with increased risk for asthma in large-scale studies. And also epithelial genes. Again, this is sort of innate immune class. You see defensin showing up here, chemokines involved with instigating the innate immune responses that then feed adaptive immune responses. So there's suggestion that susceptibility genes do play a role or are somehow involved in microbial recognition of microbial molecular patterns could plausibly be somehow involved in the pathogenesis of asthma in the context of microbiome differences. So human microbiological history, as you probably know, this is my prelude slide to basically return to the thought that asthma is more of a contemporary disease. And our lifestyles have changed over the millennia, right? So in the very early days, we were very much intimate contact with our animals, and lived in environments that are not at all, very sterile by today's standards, to an era where we are, living in much more close-knit spaces, inundated with antibiotics even early in life, and get lots of infections. And essentially, asthma is more of a modern disease, right? And we have had dramatic changes in home environments, living standards, what we eat, the exposures we have, and this all feeds into the hygiene hypothesis that you've heard about, that we've had a decrease in infectious diseases that kill us, but a rise in allergic and autoimmune diseases over the years. So this is sort of a schematic that I've had in my head over the last couple of years in terms of how I think about where microbiome fits into the asthma clinical research and translational research. There's been an intense focus looking at this end of asthma susceptibility and asthma pathogenesis is the pediatric early life literature. And those have largely focused on looking at environmental differences that are associated with asthma risk. And I'll review 1 or 2 studies in that area. The gut microbiome, which I'll touch on very briefly with one, highlighting one study in recent years. And then the rest of my talk is really going to focus on the respiratory microbiome. So the start of these very rapid review slides. So there's been an immense amount of epidemiologic literature over the last 20 years from large-scale studies in Europe as well as in the United States, suggesting that perinatal early life exposure to a diverse repertoire of microbes is associated with decreased risk of allergic diseases, including asthma. As I mentioned, there's epidemiologic studies as well as basic studies, and some of the examples are really about come from Europe, and some beautiful studies done by Erika von Muschus in Munich, who's a pediatric allergist, where she's looked very systematically at populations of kids who grew up in farms in very traditional farming environments versus kids who grew up in an urban environment. And it's very clear from data from their study, as well as other converging with other large-scale studies in Europe, that the more exposure you have in an environment associated with increased microbial richness, in this case measuring either endotoxin or the actual number of different types of bacteria or fungi found in the mattress dust of kids who live in farms in these very intimate environments, traditional Bavarian farms, is associated with decreased risk of asthma. So Parsifal and Gabriella were 2 large studies. This is a publication that came out in the New England Journal several years ago now. And basically, the more bacteria you have, the less likely you have more bacteria here, but also living on a farm is associated with increased bacterial richness, the less likely you're able to have asthma. In this other study where they looked at fungal richness in the same samples, again, that fungal richness is increased if you live on a farm compared to a cohort of children who live in the same area but not in a farming environment, so more suburban controlled population, also decreased risk of asthma over the years. And this is a figure that Erika likes to show at different meetings, showing a mother who is a farmer whose baby is basically going with her to work every day, is sitting right there next to a cow. The exposures occur very early life and even perinatally. And so that's viewed as a very critical window. And this has been replicated in other studies, including the states here, showing that exposure to dogs and cats in early life may be associated with decreased risk of asthma in later childhood. So what about the gut microbiome? So a very, very broad statement. Again, differences in the gut microbiome in early life have been linked to the risk of developing allergy and asthma. These are just a snapshot of studies. There's studies going back to 1994 using culture-based methods that have shown increased relative abundance of certain bacteria in the gut being either associated with increased risk of asthma or protective against asthma. And why is that important? I'm probably preaching to the choir a little bit, but we know that the early first year of life, in particular, the first 12 to 24 months of life are a critical window for immune development. We're born mostly sterile, we think, and the gut very quickly becomes established with microbes, and that diversity rapidly changes in the first year of life. And so anything that sort of perturbs that developing microbial system, which then trains the immune system in its development to recognize what's dangerous and what's not dangerous, is all really happening in that first 2 months of life. And so perturbations like perinatal antibiotic exposure, antibiotics given to kids, how you were born by C-section versus vaginal delivery has been associated with how that inoculum occurs and shapes the gut microbiome and subsequent immune development in the very first early of life. So it's viewed as a critical window to establishing, we think, the balance between Th1, Th2 immune functions, as well as Treg and Th17 functions. So as I mentioned, there's a lot of studies in this area. These are just some of the bacteria in the gut that have been associated with risk of asthma, such as colonization with Clostridia, B. fragilis, or E. coli, or protective probiotic species that you've heard about, like the Phytobacterium lactobacillus, either by culture-based methods in the early days or targeted PCR methods, have long been associated with either increased risk of asthma or protective against asthma. More recently, there have been these high-throughput molecular studies using sequencing methods, and I'm going to highlight one study that came out of the Canadian longitudinal childbirth cohort study, which is run by Brett Finley out of Vancouver at UBC. And this is an amazing study. They follow like hundreds of kids from basically, before they were born and up until I think 5 years of age, or maybe longer. And this is a paper that came out a couple years ago where they basically analyzed samples, fecal samples collected very early in life, starting at birth and I think at 3 months of age and onward, looking at associations between patterns of gut microbiome development using these large-scale tools and how that was associated with different disease phenotypes that are harbingers of asthma in the young cohort. So you can't really diagnose asthma in kids until they're age 6. But these terms, how they classify these kids, are ways that pediatric asthma researchers look at this. And in particular, I want to focus your attention on the atopic and wheeze phenotype. Those who are atopic and have a persistent wheeze phenotype are thought to be the predecessor to asthma diagnosis eventually in later childhood. So they noticed in their large-scale study that the atopic wheezy children later on, at 3 months of age, at very early life, already had decreased numbers and richness in these particular organisms. And I won't go into the elegant basic science translational studies where they basically took these organisms that were pulled out of an atopic wheezy child and put them into germ-free mice to see how it impacted allergic immune responses to the airways. But essentially, reconstituting these germ-free mice with these organisms decreased allergic inflammation in the lungs of the germ-free mice. But in these kids, very early on, with this pattern of decreased diversity in certain organisms, already had different profiles in short-chain fatty acids measured in their stool. And short-chain fatty acids are thought to have an immunomodulatory role, protective, and foster epithelial cell health in the gut. They also had different urinary metabolite levels already at that very early age. So and they didn't see this at 6 months of age. This was only at the sort of the very early time point samples that they saw this difference associated with later disease phenotype. Now, people have also started looking at the respiratory tract in kids. Very hard to study the respiratory tract in kids without good reason from an IRB perspective. So a lot of the studies in the pediatric realm have leveraged looking at what they call either hypopharyngeal samples or nasopharyngeal samples, usually swabs. And so there have been some nice studies coming from large birth cohorts, again, from mostly not in the United States, and I'll highlight the Copenhagen birth study in the next slide. But there's emerging evidence that upper airway microbiota, either in the nasopharynx or in the upper pharyngeal region, in early life are associated with asthma risk, suggesting that differences in the microbiome composition may modulate susceptibility to the viral respiratory infections. And viral respiratory infections, in particular with RSV or RV, have been strongly associated with risk for developing asthma later in life in childhood. So I don't have time to go through the other major studies in this area, but I'll highlight this one that you may be familiar with from now more than 10 years ago from Hans Biskar that was published in the New England Journal in 2007. I think this was the first study, not using a sequencing-based method, but using just traditional culture method. But he took hyperpharyngeal swabs from babies at 1 month of age, very early on, and then basically submitted them for traditional culture-based methods, and found that the likelihood, if you're already colonized in that space with either strep pneumoniae and Moraxella catarrhalis or Haemophilus influenza, that was associated with increased risk of persistent wheeze or asthma by age 6. And so this is a table from that study and the figure from the study that you had increased risk of developing that phenotype over that follow-up period after they initially sampled the kids. The odds ratio was pretty high here, 4.5 are being asthmatic at age 5 or 6 if you're colonized very early in life. So that window is really, really critical. And I can tell you that in the literature in that realm, the window is getting pushed even earlier, and people are even talking about the perinatal exposures and placental exposures, and that's stuff I don't do personally, but it's suggesting a lot of the factors may even predate potentially the actual birth. Okay, so I wanted to move into then presenting to you, discussing with you some of the recent evidence linking the airway microbiome to asthma phenotype in adults, and this is work that I've been involved with over the last 6 years through multi-center studies funded by the NIH and other entities. And the rationale for looking at this is, one, I'm an adult pulmonologist. We deal with patients who already have established asthma. What can we do to better understand the potential pathogenesis of certain subtypes of the disease and potential interventions and understand it better? So this is, again, a summary of how I've thought about it. And instead of looking at the microbiome at this end of the life spectrum, looking at in the study of an adult asthmatic who may have one of hundreds of different phenotypes, and how does the microbiome impact clinical features of asthma? How is it shaped by treatments that we prescribe for asthma, and how does that relate to the dichotomy of phenotypes we talk about, in particular Th2 high versus Th2 low phenotypes? So I would argue that opportunities for microbes to shape asthma span the age spectrum. So these factors that pediatric asthma researchers have focused on, looking at dietary intake, airborne pollutant exposure in kids who live close to major roadways, microbe exposures, antibiotics. These are all things that are not static, right? We continue to be exposed to those things as adults. And so why couldn't they potentially impact the microbiome and disease phenotype in chronic airway, inflammatory airway diseases? And coupled on top of that, we get comorbidities as we get older, right? So obesity, bad oral health, sinus disease, these are all, again, factors that have been associated with worse asthma outcomes in general. And then I think an area that's actually been neglected, I won't really talk about because there's not a whole bunch of literature in it, is the idea of elderly asthma. And the idea that this observation clinicians have long had of adult onset asthma is very severe, and in particular being worse in people who are elderly, defined as greater than age 55 or 60 in general. How does that play a role? And we do know that there are age-related changes in the gut microbiome itself. Could that potentially play a role in the lung disease phenotypes we see, such as asthma in older adults? So if you're not familiar with this, I thought I'd briefly go through this landmark study from 10 years ago, highlighting the relevance of type 2 high versus type 2 low asthma phenotype. And the corollary here would be eosinophilic versus non-eosinophilic asthma, which has long been recognized in the literature. But this was sort of the first study done by my colleague Prescott Woodruff at UCSF back in the day, looking at this in a molecular fashion. And what they did in their cohort was to take their cohort of asthmatic patients at UCSF and basically treat them with inhaled fluticasone, I think it was fluticasone, for I think a 8-week period, and did bronchoscopy before and afterwards. And then they also had a group that they treated with placebo. And before and they had stratified these patients beforehand based on microarray analysis they had done years prior to finding molecular genes in the epithelium that were responsive to IL-13, which is a canonical Th2 cytokine. And these 3 genes that they focused on were periostin, Clicon-1, and serpin-D2. And you can see from this heat map here that there was a type 2 high, Th2 high group of asthmatics here, and a Th2 low group here. And this Th2 low group, very much, there's A's and H's in here, so they were kind of very similar to some of the healthy controls in this particular population. What was dramatic is that when they looked at the response to inhaled corticosteroids defined by a change in lung function FEV1 after that 8-week period, they only saw a significant improvement in that lung function in the group that was Th2 high defined beforehand, and not really any response in the Th2 low group. And so this paper got a lot of press at the time, sort of validating what had been clinically observed, that corticosteroids work better against eosinophilic inflammation in asthma. And now the idea of stratifying patients by type 2 high inflammatory profiles, sort of is important, because in addition to corticosteroids, we have these immunotherapies, biologics, anti-IL-5, IL-4 therapies that have come on the market in the last 5 to 6 years that are available to treat people who have more of a type 2 high-driven process. So a lot of the criteria for treating patients with one of these drugs is, for now, based on blood EO levels of at least 150 or 200, depending on which drug you're looking at. But we really don't have any good therapies for Th2 low asthma as yet, and we also don't really understand what drives Th2 low asthma, and we have no doubt that there's many phenotypes and probably different molecular mechanisms that underlie Th2 low predominant profiles in some asthmatic patients. So now, over the last 78 years, there's been a wealth of growing studies that suggest that features of asthma associated with different airway microbiome patterns. And these come out of studies not only that I've been involved with, but with other cohorts that have been publishing in this space now, including cohorts from University of Chicago, cohorts in Australia, as well as in Denmark. And some of the clinical features that have been associated with this and that I'll go through in the next following slides include markers of airway hyperresponsive that we measure clinically, the severity of airflow obstruction, stability of asthma control, as well as obese versus non-obese severe asthma. And also responses to corticosteroids in response to macrolide antibiotics have been associated with differences in airway microbiome patterns. And of course, related to that is airway inflammation immune response patterns. And I'll just tell you that I'm going to try to convince you that both of the data so far suggests that the respiratory microbiome, at least bacterially, is more different in those who have predominantly type 2 low phenotype disease and not so much in the type 2 high population that have been looked at to date. So these two figures here, summaries of figures from two papers that came out very close to each other, as you can tell. And the first one was the very first paper we published out of the Asthma Clinical Research Network, where they were doing a parent trial looking at the effect of clarithromycin on asthma control based on whether or not there was evidence from bronchoscopy that was performed in the patients of being infected with mycoplasma chlamydophila pneumoniae. So in the chronic asthma airway disease world, there's long been this idea, and many studies in decades prior, suggesting that a certain subset of asthmatics were chronically colonized when you look for by molecular methods with either of those organisms. But macrolide studies have generally been some negative, and this parent trial from the Asthma Clinical Research Network also was some negative. But as part of that, we took the opportunity to propose a proof of concept study and look at the microbiome using a microarray tool we had at the time, look at the bacterial community composition based on something called the 16S ribosomal RNA gene. And when we looked at that cohort of asthmatics who were pretty much moderately persistent asthmatics on a standardized dose of fluticasone and compare that to the healthy controls, we noticed that the asthmatics on the whole had a higher measure of total bacterial burden. This is basically a proxy for bacterial burden where we do a 16S qPCR assay. A couple years later, around the same time, Fernando Martinez in Tucson, who is a major pediatric asthma researcher, looked at their cohort of adults at that point, and these were patients who were mostly were not being treated with inhaled corticosteroids. The more mild phenotype of asthma also showed that those who had asthma seemed to have a greater diversity. This is a common metric used in microbiome research. Seem to have a higher diversity of bacterial communities in their sputum samples compared to non-asthmatics. So between two different sample types, in this case, protected brushings that we obtained by bronchoscopy versus sputum, a suggestion that the bacterial microbiota composition differed in asthma versus health. In that same NHLBI ACRN study that I mentioned, this was the first piece of data we had suggesting that airway hyper-responsiveness as a marker of airway reactivity was associated with differences in the diversity and the composition of bacteria. And so what's shown here on the x-axis is methylcholine PC20 that pulmonologists will be familiar with as a measure of airway reactivity. So the lower the value of PC20, the more reactive, twitchy your airways are. And we saw a significant negative relationship, again, with this measure of bacterial diversity and airway hyperresponsiveness. This is a very broad metric of looking at community composition. When you dive down more deeply analytically and look at the specific types of bacteria you're getting as readouts on whatever platform you use, we saw that this relationship was driven by airway enrichment, increased abundance, if you will, in members of this phylum called Proteobacteria. And if you haven't heard that before, I'll just keep that in mind. Proteobacteria is a very large phylum, but it encompasses a lot of the bad actors that we think about in respiratory disease as well as in GI disease. So it includes Haemophilus, it includes Klebsiella, Moraxella. In the gut, it includes Enterobacteriaceae, E. coli, et cetera. It's a very big family, phylum of organisms. We then went on towards the end of that study to be involved with another multi-center study that was being conducted by Genentech at the time, looking at markers of steroid-resistant asthma in a severe asthma cohort. They recruited across something like 20 centers in the U.S. This was all very severe asthmatics, so even more severe than the prior cohort that I showed you from the ACRN, and they were all poorly controlled despite being on high-dose inhaled corticosteroids. They did a very simple observational study over approximately 4 to 6 week period where they brought them in, characterized them deeply by clinical questionnaires as well as getting samples, including bronchoscopy and sputum. And we again proposed to them, hey, we've done this before with a less severe cohort. If you've got brushes from this cohort, with the same sample type, we could take a look at that and compare it to the other cohort. And that's exactly what we did. Importantly, within this observational study, they were not allowed to have any clinically significant changes during the study, and no medication changes. And so again, we did an exploratory analysis with all the metadata, clinical and inflammatory, that they had collected. So what I'm showing you here is something called a non-metric multidimensional scaling plot. And it's a big word, but basically it means distilling down lots of different information into a point and mapping them out. So each of these points here, if you've ever seen something like a principal components plot, it's very akin to that, just a different statistical methodology. Each point basically represents the sum bacterial community in the bronchial brush of a given subject. So we analyzed about 30 out of 40 subjects in this study. And what's superimposed here are these vectors, and these are what we call gradient vectors. And what's labeled here are some of the variable names from the metadata that we looked at that came up as being significantly associated with differences in community composition. So the direction of the vector gives you a sense that maybe some of these factors associated with, say, differences in asthma control, defined by a questionnaire we use in asthma clinically called the Asthma Control Questionnaire, or a number of leukocytes in the sputum, might be associated with a slightly different community composition compared to, say, body mass index in this cohort. And that's what I'm going to dive into a little bit more in the next couple of slides. So out of all the variables that we looked at, essentially, we saw associations between differences in bronchial bacterial community composition, microbiota composition, and stability of asthma control in this population over a 1-month period, obese versus non-obese patients in this cohort, and markers of steroid responsiveness or type 2 inflammation. So this is the data from the obese versus non-obese severe asthma patients in this cohort. And when we looked, basically, separated them into those two groups and looked at what differed between the groups in terms of the effect, in terms of their bronchial bacterial community composition. The obese severe asthma patients were significantly enriched in quite a few organisms and these two other phyla called Bacteroidetes and Firmicutes. And these are organisms we hadn't up until this point really thought about in our disease. I mentioned proteobacteria before. And so this was really kind of striking to me at the time when we came upon this in our analyses, because these are the two dominant phylum of organisms in the gut. And these are a lot of anaerobic organisms. And so it suggests to me, and follow-up studies have yet to be done in terms of looking at the function of these, but for some reason, the obese severe asthma patients in this particular cohort seem to be enriched in some of these anaerobic organisms more associated with the GI or oropharyngeal cavity. When we looked at asthma control within that 1-month period, so in the design of their study, they had assessed asthma control within a 4-week period using the ACQ questionnaire. Again, they had no clinical changes detectable by the patient, but yet at the same time, a subset of patients seemed to have indicators of worsening asthma control. So a positive change in this ACQ metric indicates worsening asthma control, and a 0 score, 0 change or negative change on this particular questionnaire indicates improved asthma control. And basically correlated, what was associated with those changes in the direction of that score. And again, overwhelmingly, the organisms that were strongly positively associated with indicator of worsening asthma control, at least less stable asthma control, were mostly this proteobacteria group of organisms. Over 90% of the bacterial groups that we identified were correlated with that. Whereas the converse, a very different bacterial group called actinobacteria were correlated with evidence of stable or at least improved asthma control. And I highlight this in blue and red here because they're very different phylum of bacteria. Proteobacteria, as I mentioned, represent a lot of potentially pathogenic organisms, at least in the respiratory tract as well as in the gut. Actinobacteria, not so much. They're actually more encompass organisms that include the environmental organisms. They're very prolific makers of secondary metabolites. So we get antimicrobial compounds that we use like streptomycin, neomycin come from members of the actinobacteria class. So just metabolically very different group of organisms. Now in this study, our collaborators at Genetic had done epithelial gene expression profiling, and so gave us some information on those gene expression readouts to look for associations with the microbial readouts we have profiled, at least bacterially. And when we looked at, out of the 4 gene expression signatures they gave us, which included a Th2 gene signature, like the one I showed you a couple of slides ago, this pattern, which they define as indicative of steroid responsiveness, basically expression of FKBP5, which is involved in the glucocorticoid receptor engagement, as well as a steroid-resistant pattern, mostly defined by IL-17 responsive genes. So IL-17 pathways have generally been thought to be more steroid-resistant in terms of their treatment response in patients. And when we looked at these two signatures, were the only two out of the four, not the Th2 one, but these two signatures came up with significant associations with members of the bacterial community, and that's shown here in this plot here. So what's shown in F here are the organisms that were significantly associated with this steroid-responsive pattern, and what's shown in red here and overlaid with sputum neutrophilia, which kind of goes with the Th70 IL-17 neutrophil chemotaxis type pattern, were associated with a very different group of organisms. And what were the organisms, what were the phyla that these organisms belong to? Again, proteobacteria and actinobacteria. And so remember on the previous slide that the actinobacteria seem to be associated with more stable or improved asthma control, whereas proteobacteria, again, were associated with more less stable control in the previous slide. So it seemed to dovetail nicely with the clinical readouts we were showing on the previous slide. So I mentioned that the Th2 signature, surprisingly to us, we didn't see any signature associated with that. We looked high and low, looked at every possible metric, including sputum eosinophils, biopsy eosinophils, et cetera, and just really didn't see any correlates from the bacterial community, at least, and markers of type 2 inflammation. And what we noticed is that when we went back and looked at these data, as I mentioned, we noticed that those in whom we could look at the bacterial component of the airway microbiome, those patients in whom we could do that profiling had significantly lower sputum eosinophils, seemed to have lower biopsy numbers, and when we actually did a quantitative PCR looking at bacterial burden in those samples, there was a negative relationship between that, the number of biopsy eosinophils taken from these patients. So we started to have this emerging feeling that maybe the bacterial community differences we were seeing in the airway microbiome might be associated more with a non-type 2, type 2 low inflammatory profile in the lungs. So a question we would always get up until this point, we were designing the study at the same time, is all these patients were on inhaled corticosteroids. What is the effect of inhaled corticosteroids, which are prescribed so widely in pulmonary medicine? And so many years ago now, we set out to design a very simple proof of concept study within the NIH AsthmaNet Consortium. We started this when I had my K award, and we're still analyzing data from it. And the design was very straightforward and descriptive. And basically, we thought we'd enroll 3 groups of subjects: allergic asthmatic, atopic asthmatic subjects who really had not required inhaled corticosteroids for very mild asthma clinically, and enroll 2 groups of control subjects, non-atopic, non-allergic healthy controls, as well as a group of people who had allergic sensitization to aero allergens but didn't have evidence of asthma that we tested for in the trial. And then we put them through bronchoscopy as well as sputum induction, collected samples, and then within the asthmatic group, we randomized them to inhaled fluticasone versus placebo for 6 weeks and studied them again afterwards. Collected a bunch of specimens that allowed profiling of immune responses, as I mentioned here. And just to give you a snapshot of some of the findings, again, most of these subjects seem to have type 2 low profiles, which kind of surprises a little bit, but at least by the way we approach defining type 2 inflammation study, again, based on expression of these IL-13 inducible epithelial genes, only about 10 of our 40 asthmatics seem to have a type 2 high profile. So most of the data, again, focusing on the bacterial community, seem to be focused on the non-type 2, type 2 low population. That's capitulated here. Again, when we looked at bacterial burden on the y-axis between the type 2 high versus the type 2 low subjects, significantly more in type 2 low subjects, although admittedly there is a spectrum here, and I think that gets at the heterogeneity within the type 2 low population. When we looked at the bacterial communities, and again, in the protective brushings we collected at bronchoscopy from these subjects, and plotted them out and compared them side by side, this slide summarizes that. So all I'm showing here, the names aren't really that important for the purposes here, are all the bacterial groups defined by sequencing that were significantly associated with either the asthmatic group or the atopic group. And what's shown here in the columns are the groups, bacterial groups that we think are strongly associated either with asthma or either with atopic status. And we reasoned that when we did the 2-by-2 comparison of comparing the asthmatics to the healthy non-allergic group, as well as the asthmatics to the allergic but non-asthmatic group, that if you saw blue bars or red bars across both of those comparisons, that the likelihood of being more strongly associated with asthma, the evidence was stronger there. And that's what's highlighted here. And that certain organisms like the fusobacterium, which is an anaerobe, were strongly associated with asthma in both of those comparisons, as well as a member of the Haemophilus group here, there's some names here. Whereas Prevotella, again, usually an oral associated organism, but we're finding more and more of it in lung studies. Seem to be more associated with atopic only pattern. And so if I flip back, you get the sense that the atopic only group, the organisms that were associated with atopic status without asthma, seemed just as striking. And that was a little surprising to us, but we thought to be an important control group because so many asthmatic individuals are allergic. Is there something different about being allergic versus non-allergic in asthma? And so from this data, we would submit that controlling for atopic status, at least in airway asthma studies, is likely important because we don't really know what the effect of atopic sensitization is on the microbiome, although people who study food allergies and the gut microbiome have reported differences in the gut microbiome. So I think there's some evidence to lean on there about whether atopic sensitization predisposes you to being colonized by organisms more readily. When we looked at responses to the inhaled corticosteroids, so here's the caveat of doing bronchoscopy studies for microbiome science in large scale, is that it was very hard to get paired samples from patients before and afterwards. It's just differences in operator, collection of samples, patients and cells, even in a multi-center study. So we didn't feel very confident in showing some of the pre and post data, but we did look at the baseline community differences in those who seemed to show response in inhaled corticosteroids, even though they were mostly type 2 low, versus those who did not respond. And we define inhaled corticosteroid responses as an improvement in bronchial reactivity by this clinical test. When we looked at the baseline pre-inhaled corticosteroid delivery to these asthmatic subjects, we noticed that at baseline, those who seemed to show an improvement in their methylcholine PC20 seemed to differ at baseline in their bacterial community composition compared to those who did not. The y-axis here, without going to great depth, is basically a readout of how the different communities are between comparator group, in this case, comparing it to the reference group of healthy controls. And there's a difference there between those who did respond to steroids versus did not respond to steroids versus not. And then when we use a bioinformatic tool that's out there to basically try to infer what bacterial communities can do metagenomically, what kind of genes are potentially encoded in their genome, a lot of the pathways that came up in the database seem to be associated with metabolism of xenobiotics and drugs and external compounds, suggesting, at least to my mind, the possibility, and admittedly it's speculation, it's still at this point, that there's something different about the community and those who can't respond to steroids, and those communities seem to be enriched in gene content that might be associated with being able to metabolize xenobiotic compounds. Okay, so here's a summary of sort of the data to date. So, we've looked at this, the role of the respiratory microbiome in the setting of type 2 high versus type 2 low inflammation. I hope I've convinced you, at least with the data we currently have on hand, that when we look at the bacterial component of the airway microbiome, that there seems to be more of an association between differences in bacterial community composition in the airways and type 2 low inflammation as opposed to type 2 high inflammation, which I think is really interesting. And we think we see an effect with inhaled corticosteroids as well. There you go. Okay, so in the time remaining, I thought I'd discuss some of the challenges and new directions to understand how microbiota influence asthma phenotype. So, as I mentioned with a previous study from the NIH AsthmaNet, it's terribly hard to do a bronchoscopy. And although I don't have time to go into some of the data we've more recently generated looking at different specimen types, including sputum as well as nasal brushes, it's really a sort of a hindrance to doing longitudinal studies. And I think it's really important in chronic airway disease to be able to do longitudinal studies if you're looking to look at disease phenotype as well as the microbiome. And that really hasn't been done because this is really hard to do longitudinal studies using to re-bronch patients and convince patients to participate in studies where we're going to do repeated bronchoscopies on you. And so I think there's a need in our field to sort of move and try to validate other specimen types. In particular, I think induced sputum is promising, but is also much more complex. On the other hand, there's a lot of background in asthma and COPD research validating the use of looking at induced sputum and immune inflammatory markers and how they relate to disease outcomes in those diseases. So somehow I think it'd be nice to be able to leverage looking at the microbiome represented in induced sputum to infer lower airway microbiome composition that you might otherwise obtain by brushes or lavage. But really, we have no idea of the stability of the microbiome in chronic respiratory disease, with the exception of cystic fibrosis, where there's been a lot of literature in this over the years. Even in cystic fibrosis, which is a unique clinical context, it's pretty stable. But yet, when they have exacerbations in CF, they do see a perturbation in some patients, but not all, in their microbiome. But we really have no context for understanding that in the setting of asthma. There's a little bit of data of this in COPD, but there's a lot of work to be done there. And I think that's terribly important because if we think that microbiome information might be incorporated into phenotyping schemes to better understand the phenotype of a patient, I would argue we kind of need to understand what foundation we're working on. What's the baseline background stability? If you're going to develop a predictive marker, how does that compare to the background variation over time in a predictive model is sort of important to know. Also, the functional implications of microbiota differences. How are they linked to asthma phenotype? And this is a huge area in and of itself. Even people who study the gut microbiome wrestle with and tend to sort of focus on individual species, how to get at that and understand how a specific microbe functions, let alone in an ecosystem of other organisms that they are living in the same neighborhood as. And so the challenges here are complexity at two levels: complexity in the microbiome level, diversity, strain-specific or species-specific functional diversity, and all the data. It's an immense challenge, and I would say even those in the gut microbiome space who've been doing this for decades, I think, still struggle with this challenge. And we've also overwhelmingly focused on bacteria and largely ignored fungi and viruses, what people might call the microbiome or the virome. And they come in and of themselves with challenges in studying because how you process those samples, look at those elements of the microbiota are different. And when you're working with something other than stool, which is more readily to get, it becomes very hard to make decisions up front about how you're going to take your sample, process it for the different purposes you'd like to do with that. And then couple that with the patient phenotype, which is very different from a mouse phenotype in that there's so many more factors to look at, so many more elements that can shape the microbiome. And I won't go through all this, but we certainly know that, what the transcriptome and what these gene polymorphisms actually do in terms of conferring susceptibility to asthma is still not quite understood. And then aspects of epigenetics and environmental exposures that might further modulate host immune responses. How does that all integrate itself with the microbiome and other readouts we get of how the host is responding in the disease phenotype? So really, in the space of the respiratory microbiome research, we've largely focused on microbiota composition, understanding who's there and what's different. And it's descriptive, it's very basic, but it's basic stuff that has needed to be done and probably still needs to be done in the context of large population studies where we look at different disease phenotypes. So you really can't capitulate, I think, as well in mouse or animal models. And people have started to look at function, try to infer what these microbial communities can do using in silico tools developed by computational biologists and engineers, which are helpful. But ultimately, we need to sort of be moving more towards integrating, if you're an omics type person, leveraging these different technologies and different platforms that measure different readouts of what the system puts out. And so one area that we're focusing on with the cohort that I'm building at Michigan is using metagenomics, focusing on the microbial community to understand what are the genes encoded by these microbes and what they potentially can do. And eventually, hopefully, coupling that with metatranscriptomics, the mRNA, the gene expression readouts from the microbial community, as well as these readouts of the actual protein products or the metabolic products to actually mediate these interactions. So as Janet Jansen, who's a big environmental microbiome scientist out at in Washington State, published in this journal, is really talking about the phenome, the phenotype, if you will, of a microbial community, and how does it interface with the phenotype of the human disease context. I would say that this cannot be done in isolation because to really understand the mechanisms, we still need to continue applying traditional experimental methods, microbial culture work, in vitro, in vivo work, leveraging patient samples to really bring it all together. So summary, some 3 points I hope to leave you with is that I hope I've convinced you that lower respiratory microbiome asthma, as well as in chronic airway diseases, which I didn't have time to show you data from, differs from in health, but seems to evolve in parallel with disease advancement. And in the clinical context of study, it's very hard to tease out what's chicken and the egg. Is it the severity of disease that's driving the microbiome changes or vice versa? And I think it's probably no doubt bidirectional, but it's difficult to dissect that in a clinical context. Microbiota association's clinical features in asthma seem to implicate microbial influences on asthma phenotype, but as I mentioned, the bidirectionality of these links needs to be further studied. But I think it's really intriguing and maybe give some weight to some of the people who, in decades past in the pulmonary world, really felt like macrolides helped a certain subset of population, subset of patients. But how do you actually identify those patients and define them to say who's going to most likely benefit from a microbiome or microbiota targeted intervention? I still think foundational studies are still needed to address microbiome disease phenotype relationships in different sample types. We've started to do this with induced sputum as well as nasopharyngeal samples. They're very difficult samples to work with, but it'd be nice if we're going to eventually, in the future, try and move this into a clinical realm where you could take a sample and understand what the microbiome is and how that relates to the clinical disease phenotype or your blood eosinophil level, et cetera. We need to be able to do this outside from convincing someone to undergo a bronchoscopy for a clinical indication. And functions elaborated by human microbiota, impact lung disease, as I mentioned, is still very basic stuff that needs to be done that will take another generation probably. So with that, I'd like to end and thank you for inviting me, some members of our asthma and COPD microbiome research group at the University of Michigan and collaborators at UCSF and senior investigators at Michigan, including Gary Huffnagel and Yvonne Huang. And as always, grateful for my funding to allow me to do stuff that's still fun to me. So thank you very much. Thank you.
Delightful talk. I guess the major message that I take away from the human body is nothing more than a bunch of
Microbes maketh man.
organisms. So let me start off the questions with asking you, is there any evidence that composition of your microbiome is influenced at all by blood type?
Great question. I don't know that. I'm not familiar with any studies that have looked specifically at blood type, but you raised interesting related sort of body of evidence where people have looked at blood type and susceptibility to viral infections in the gut, such as susceptibility to norovirus infections. John Fye at UCSF several years ago published data looking at susceptibility to viral respiratory asthma exacerbation associated with presence of the H antigen on, I think, epithelial cells we looked at. So there's connections there with susceptibility to being infected by viruses. I will say that there has been a study that's attempted to look at this in the bronchiectasis realm, and I believe that study also seemed to show an association would be more likely to be colonized with Pseudomonas and non-CF bronchiectasis, the particular blood types, I forget which one. So people are starting to look at that because that data is there and being able to do Lewis antigen typing as additional metadata to look for correlates is there, but there's not been a ton of studies yet in that realm. Mm-hmm. Right. So EBC, exhaled breath condensate, is a sample type we can collect. I can tell you that with attempts to analyze that sample type to look at DNA or microbiota composition, very difficult, and we really don't see that. There's just not enough cells and DNA there. But to answer your question about metabolome, people have looked at the metabolome of the lung using lavage fluid to start because they've collected it from their bronchoscopy-based studies. I will tell you there's debate in the field, at least about sort of how you analyze that metabolic data in a sample that's you create and how you normalize that. And so there's debate about how you go about that. But people have attempted to measure metabolites by GC, MS or LCMS from lavage fluid. But I'm not familiar with other specimen types just yet. Mm-hmm. Yeah, I haven't seen data where people have looked at exhale breath patterns, like volatile VOCs and stuff, and correlating that with microbiota. That'd be interesting. We are going to incorporate that in the study that we're doing at Michigan, where I've linked up with a biomedical engineer who has an exhaled, we basically could collect exhaled air and apply it to a portable LCMS platform that he has to look at, real-time readouts of that breath. So down the road, we hope to be able to do that if we get that up and running. It's possible, yeah. Although, there's robust literature in the asthma world looking at either EBC, exhale air, or blood metabolome. And so I think the start is to be focused, and my goal is with this platform is to first focus on those compounds that have been published in literature that seem to have been associated with asthma or asthma exacerbations, and go from there. The actual source, whether they're truly from the lung versus oropharynx, I'm not sure that's been well determined. Yes. So in the adult world, there hasn't been that much done yet. We are starting to do that. In fact, at our upcoming meeting, I'm going to present some data from our cohort. We've looked at the gut microbiome, the subset of our cohort in Michigan, and are seeing some intriguing patterns between gut microbiota composition and markers of asthma, including lung function. It's very preliminary data. I think that one of the challenges, admittedly, is that so many things can influence the gut microbiome than established in literature. And I think, from an analytical standpoint, how you dissect the relative contribution of those other factors that can associate with differences in the gut microbiome, and you can't control for everything, but trying to control for them in as much as you can. But I think the story where this may more readily flesh out sooner would be the obese asthma story, because obviously obesity has been associated with many different things, including the gut microbiome, systemic markers, inflammation, and almost certainly it's got to be a bottom up influence on susceptibility to lung disease and asthma-like phenotype. Mm-hmm. We have not. Trying to think if we haven't even actually, at least in the asthma world, started to look. I've made attempts to actually try to measure short chain fatty acids in blood in the lungs. We've run into some technical difficulties associated with that, to be honest. I will say in that figure I showed you of the data from AsthmaNet, where we tried to infer some of the bacterial community functions from those organisms that were associated with asthma, that the short chain fatty acid pathway came up. So it's of interest, at least when I've talked to the people who work in the metabolomic realm and talked about trying to measure short chain fatty acids in something other than the stool. There's concerns about sort of what the levels are, if the assays are sensitive enough. We'd like to, but we haven't done it yet outside of the gut.
Thank
Thank you.
Talking for all of our guests.
Oh my goodness.
We lead the Louisville Slugger, which has your name, Yvonne Huang, Medicine
Yes.
Grand Rounds date. Thank you very
so
much.
much. That's wonderful. Yeah, thank you. What a surprise. Thank you.
Now, okay, so Jason is
Okay.
I
can take this on the plane.
No, you can't take it on the plane. No, we'll ship it to you.
Thank you.
We'll ship it to you. So that's
Yeah, I don't think so. There it is. That's cool. So you've been at Louisville most of your career, all your career?
most of my career, 27 years I've been here.
Hi.
Yvonne. So we do fecal metabolomics, and
Okay.
so like I say, a lot of the stuff that you see in asthma, we see in feces, either severe alcoholic.
Yeah.
And are alcoholics more predisposed to asthma? Has anybody looked at that?
I haven't heard that,
Yeah.
but I will tell you, in the preliminary data we have from the gut, where we've done very extensive dietary questionnaire, and I think the readouts we're getting from this questionnaire are good, but alcohol actually